Fundación Cana brings you the following article. Medical cannabis. Would using the plant have advantages over using the individual ingredients? By Dr. Carlos Goicoechea, Dean of Pharmacology at the Rey Juan Carlos University in Madrid, and Dr. Nuria Acero, Dean of Pharmacognosia at the CEU San Pablo University in Spain. Recently, the Spanish Parliament's Health and Consumer Commission has endorsed the regulation of medical cannabis in Spain. This is a pioneering initiative both in terms of how it was carried out and due to the fact that the possibility of regulating the use of a plant rather than an active ingredient is being considered. One of the most frequently asked questions about cannabis and its medicinal use is whether it is preferable to use medicines made from the active ingredients of the plant or their synthetic derivatives, or to use the plant itself. Cannabis is an annual herbaceous plant and it is dioecious, i.e. the male and female flowers are found on different plants. The part of the plant that is used therapeutically and also in recreational use are the female flowers, which are arranged in inflorescences located in the axils of the leaves. The female flowers are protected by bracts, modified leaves, on which a large number of hairs can be observed. The active principles are found in these hairs, chemical compounds responsible for the pharmacological activity of cannabis. Chemically, we can highlight the presence of three types of molecules: cannabinoids, terpenes, and flavonoids. Cannabinoids, and especially tetrahydrocannabinol, THC, have been extensively studied since 1964. Other cannabinoids can be also found together with THC, cannabidiol, CBD, cannabigerol, CBG, cannabidivarin, CBDV, or tetrahydrocannabidivarin, THCV. These appear in the plant in the form of acids that are transformed by heating or combustion into the aforementioned compounds, which are the ones that really impact on our organism. The amount of concentration of the various cannabinoids differs from one strain to another, but since THC is the one that has a psychotropic effect, the levels of this compound in the strains used for recreational use have been increasing over the last 30 years. Is the same thing happening with medical cannabis? We will provide an answer later on. Many diseases are multifactorial, which means that the activation of different receptors may be required to achieve a therapeutic effect. Plants and their extracts, possessing complex mixtures of active principles, can achieve this objective better than a single synthetic compound, as the components can act synergistically. The presence of multiple compounds in the same plant has given rise to the development of a concept known generally as the entourage effect, which refers to the possible synergistic effects between the different components, in this case cannabinoids, of the plant, allowing for the improvement of its therapeutic capacities with respect to the isolated components. In this case, the effect would cover both cannabinoid-cannabinoid interaction and cannabinoid-terpenoid or cannabinoid-phenol interactions. Combining THC and CBD. The pharmacological effects of THC and CBD have been extensively studied. In short, THC has been shown to have psychoactive, anti-inflammatory, anti-cancer, analgesic, muscle relaxant, neuroantioxidant, antispasmodic, and bronchodilator effects. For its part, CBD exhibits anxiolytic, antiemetic, antipsychotic, antiarthritic, analgesic, anti-inflammatory, anticonvulsant, and immunomodulatory properties. So far in Spain, we only have 2 drugs made with cannabinoids. One of them is made with a mixture of THC and CBD and is indicated for the treatment of spasticity and pain in patients with multiple sclerosis and for the treatment of opiate-resistant pain. The other, with CBD only, is used mainly for the treatment of seizures and epilepsy in children, although its usefulness in the treatment of chronic pain is currently being studied. One of the major advantages of these drugs is that we can precisely control the concentrations and dosage of the active ingredient administered to the patient, something that is much more complicated if we work with a medicinal plant as a whole. Despite the interesting therapeutic effects of THC, these are inevitably linked to undesirable side effects such as memory problems, attention problems, cognitive alterations, motor coordination difficulties, psychosis, including schizophrenia, tachycardia, hypertension, and xerostomia. However, it has been shown that the joint administration of THC and CBD allows the appearance of joint effects, synergistic and antagonistic, due to the entourage effect. This would allow CBD to reduce the adverse effects of the former, such as psychotic symptoms and cognitive impairment. Some studies, such as that of Johnson et al., add relevant data in this regard. These authors conducted a clinical trial comparing the efficacy of an extract with THC alone, 2.7 milligrams per 100 microliters, and another with THC and CBD, 2.7 and 2.5 milligrams per 100 microliters, versus placebo in the treatment of pain in 177 patients with advanced cancer, showing that the mixture of the two cannabinoids achieved better efficacy and tolerability results than THC or placebo alone. These and other results point to a synergistic effect of both compounds, which would achieve a greater therapeutic effect with lower doses than those required when the active ingredients are administered separately and therefore with a lower incidence of side effects. In view of these data, it could be extrapolated that likewise, the use of the plant as a therapeutic remedy by providing both compounds and many more would result in a reduction of side effects and therefore a greater acceptance of the treatment by the patient. In this sense, as mentioned, CBD would help to reduce side effects of THC such as anxiety, sedation, cognitive deficits, memory deficits, or hunger. The optimal ratio of CBD and THC in the plant or in the drug varies according to the pathology to be studied and is even different depending on the patient. Despite all this, the required concentrations of THC in medicinal cannabis, less than 10%, are usually lower than those in recreational cannabis, less than 15%. The combination of both active ingredients reduces side effects, but we can also surmise that it also improves tolerability. There are several studies that support that CBD and perhaps other cannabinoids present in the plant can act synergistically with THC, enhancing its beneficial effects, improving its therapeutic capacity via a multi-targeted effect, reducing adverse effects, and improving some pharmacokinetic aspects such as the solubility or bioavailability of its components and/or its metabolism. Do terpenes contribute? As mentioned above, the effects of isolated cannabinoids as well as their synergistic effects have been studied, and we can find abundant literature on the subject. However, apart from THC or CBD concentration, certain patients showed a preference for strains that had specific concentrations of some terpenes. Terpenes are molecules that can accumulate in the glandular hairs of the bracts of the female flowers, and that fundamentally gives the plant its smell and flavor. They are a fundamental component of essential cannabis oil, and more than 100 of these compounds have been identified in the plant. The question that arises is whether this preference on the part of some patients is influenced only by these organoleptic characteristics of the plant, or whether there is really some type of therapeutic benefit associated with the presence of these molecules, both in quantity and variety. Today, we know that these compounds are also pharmacologically active and that together with cannabinoids, they constitute the main secondary metabolites of cannabis, without forgetting phenols, which we will discuss later. Among the most abundant monoterpenes and sesquiterpenes, we can mention limonene, alpha-terpineol, camphene, linalool, camphor, alpha-pinene, beta-pinene, beta-caryophyllene, and myrcene. These compounds are not exclusive to cannabis, and they are also found as part of the essential oil of other plants. Their pharmacological potential has been analyzed through preclinical and clinical studies, either with isolated terpenes or through essential oils with high concentrations of some of these compounds. For example, in laboratory animal studies, limonene showed anxiolytic, analgesic, anti-inflammatory, and antidepressant effects. This molecule contributes to muscle relaxation and aids sleep. Beta-pinene increased gastrointestinal motility, while alpha-pinene is an inhibitor of acetylcholinesterase and therefore could improve memory. linalool showed analgesic, anticonvulsant, and anxiolytic activity, while myrcene showed anti-inflammatory, analgesic, and anxiolytic activity. Beta-caryophyllene binds to CB2 receptors and displays anti-inflammatory and gastric protective activity. Therefore, terpenes show a multitude of pharmacological actions, and as there is a great variety of these compounds in cannabis, it cannot be ruled out that the aforementioned entourage effect related to the synergy of antagonism of cannabinoids and terpenes could explain the multi-target pharmacological effect. In addition, terpenes may also affect some pharmacokinetic aspects of THC, such as enhancing its bioavailability, the amount of substance reaching systemic circulation, through an increase in blood-brain barrier permeability, modulating the affinity of THC for the CB1 receptor, and interacting with receptors for certain neurotransmitters, which could explain how terpenes modulate the analgesic and psychotic effects mediated by cannabinoids. Let's not forget phenols. As for phenolic compounds, we can highlight the presence of different flavonoids, especially flavones and flavonols, such as apigenin, luteolin, camphorol, or quercetin, which are found both free and in the form of glycosides. In addition, there are two flavones exclusive to this plant, canflavin A and B. Lignans and stilbenes are other phenols present in cannabis. Phenols in general are antioxidant compounds that help prevent numerous pathologies such as cardiovascular diseases, neurodegenerative diseases, or cancer. These types of molecules are not only able to scavenge free radicals, especially reactive oxygen species, but also modulate the levels of cellular antioxidant enzymes. Thus, for example, apigenin is anxiolytic and together with other flavonoids present in cannabis shows a mild estrogen effect, while on the other hand, canflavin A is a potent inhibitor of prostaglandin E2 and of cyclo and lipoxygenases, thus producing an interesting anti-inflammatory effect. Interactions of cannabinoids with other plant components. Scientific evidence of cannabinoid-terpenoid interaction is scarce. Information mostly comes from dispensaries extolling the virtues of patented chemical strains of cannabis or chemovars. However, as we have seen above, some terpenoids have intrinsic physiological effects, so the idea of a combined effect should not be ruled out. In a survey of nearly 2,000 patients using medical cannabis, those with migraine preferred strains with higher concentrations of beta-caryophyllene and myrcene. This could reflect how the documented anti-inflammatory and analgesic properties of beta-caryophyllene and beta-myrcene could be coupled with the potent analgesic, anti-inflammatory, and antiemetic properties of THC. However, again, we found contradictory results. In research conducted to analyze the contribution of terpenes and cannabinoids in cannabis-mediated analgesia in rats, which were intraperitoneally administered cannabis extract with and without terpenes, isolated terpenes, and delta-9 tetrahydrocannabinol, THC, no differences were found between rats treated with the whole extract and extract without terpenes, and it was further noted that the extract containing only terpenes was not active. According to studies conducted by Santiago et al. In murine cell cultures transfected with cannabinoid receptors, none of the most abundant terpenes in cannabis can affect the interaction between THC and CB1 or CB2. This does not rule out the possibility of an entourage effect, since this interaction could occur by other means, such as acting on other proteins, affecting the metabolism and distribution of cannabinoids, interacting with G-protein-coupled receptors, or with ligand-activated ion channels, among others. There are many other ways in which these molecules could interact with cannabinoids to influence the overall therapeutic and subjective outcomes of cannabis administration. We have reports of consumers with clear preferences for certain strains of cannabis, which could be explained by the entourage effect. However, these are personal experiences, and we need scientific evidence. Researchers agree that THC is the compound with the greatest therapeutic value, but the use of the plant as a whole can be equally valuable. More studies are needed because, for example, we do not know in what concentration terpenes reach the central nervous system and the brain when cannabis is consumed. Studies are needed in which different types of cannabis are analyzed with different concentrations of terpenes and phenols, since for the most part, the effect of the sum is studied by simultaneously administering various previously isolated compounds. It is difficult to find works in which a pharmacological analysis of various types of cannabis is combined with an exhaustive phytochemical analysis of the samples used, which makes it difficult to extrapolate the findings to the consumer's habitual usage. Do the effects of medical cannabis vary according to the modified varieties, cultivars, and the manner of consumption? The cannabis plants from which the various medicinal preparations are obtained can show important differences in the proportion of cannabinoids, but also of terpenes and/or phenols. The final composition may depend to a large extent on processing conditions, for example, the evaporation rate of some components during these steps. Many of the available medical cannabis preparations, especially oils, show lower amounts of terpenes, especially monoterpenes, than the plant, while this amount remains higher when cannabis is consumed by smoking, which could explain the preference of some consumers for cigarettes or vapes versus oil. Additionally, given the existing legal situation, consumers, patients or otherwise, do not usually have a single source for the product, which makes it even more difficult to obtain reproducible effects. The forms of consumption also influence the availability of the different components. For example, if cannabis is vaped, terpenes are inhaled before cannabinoids, although the impact that these differences in absorption may have on the pharmacological effect is still unknown. On the other hand, in most of the studies, the composition of the different preparations of the plant is not detailed, being limited to expressions such as cannabis extract or cannabis oil, which does not allow one to know the concentrations of the different components. Differences in processing and consumption methods make it extremely difficult to know, let alone predict, the importance of the effect of the various plant components on the final effect of medicinal cannabis. Therefore, we could truthfully say that there is not a single entourage effect, but rather innumerable ones, depending on the presence and proportions of the various compounds due to the different forms of consumption. In summary, the use of the cannabis plant allows the joint action of several of its components, which could reveal beneficial effects that have not yet been scientifically demonstrated. The cultivation of specific varieties under controlled conditions, humidity, temperature, soil, hours of light, etc., together with standardized systems of extraction, purification, etc., would also allow products with stable concentrations of their different components. Thus, known and controlled compositions of terpenes, phenols, THC, CBD, and other cannabinoids as a whole would give us the opportunity to profile more effective drugs personalized for each patient's needs. Undoubtedly, taking advantage of the entourage effect would increase the pharmaceutical value of cannabis and justify the use of the plant over its isolated components. This article was brought to you by Fundación Cana.